afternoon or good morning, depending on where you're joining from. I'm Dr. Anna Schwabe, and I'm not going to talk much about research today, mostly because I haven't been in the lab in quite a while. Instead, I want to focus on language and the disconnect in how different parts of the cannabis space communicate with each other. So scientists, industry, and consumers use different vocabularies, and when those don't align, it creates confusion, miscommunication, and potentially broken trust, especially when the common language doesn't match scientific terminology. So I want to explore today how we got here and how we can start building a better shared way to talk about cannabis. So here's a quick overview of what I'll be talking about, and I'm going to be going super quick so I can make it through in time. But I'm going to talk about why language matters, current categorization schemes, what's not working, what science shows, and then hopefully a call to action to build a better communication system. So in cannabis, we have, I feel, a slight communication problem. Here I have a list of multiple ways that we can and do categorize cannabis. And you can probably look at this list and realize there's something missing or misguided in each one of these classifications. None really encapsulate everything that's needed to communicate effectively about cannabis. So we've got cannabis and cannabis, and unless it's in writing, they sound the same, but they do have different meanings. And we go through the list of the indica sativa thing, the hemp and marijuana thing, the strains versus cultivar thing. We've got terpene profiles, and as we know, terpenes are volatile, and they may not be as relevant as we think for combusted flower. And nothing in nature fits neatly into a box. So that's something to think about. But for example, here are 4 different papers, all using different language for cannabis categories. My paper up here in the top left, I used the terms drug type and hemp type. And I debated using drug type because CBD is technically a drug too, but in the US, CBD dominant cannabis is usually classified as hemp. So, but we wanted to avoid using the term marijuana due to concerns at the time over its racist history. Then we have broadleaf marijuana and narrowleaf marijuana that the paper up in the top right used, along with hemp and hybrids. The one in the bottom left used marijuana and hemp, and then the one in the bottom right used type 1, 2, and 3 based on their dominant cannabinoids plus hemp and landrace. So we've got 4 different categorization systems that lead to confusion, not just among scientists, but also in how we communicate with the public and in the industry. And I'm not calling anybody out. I just saw this particular example while I was reading through this paper. And it's funny that this section is titled inconsistent naming because they use both cultivar and strain in the same short piece of work to describe the same thing. And it's this mixed terminology that's quite common across cannabis research and industry writing, but it highlights how language isn't standardized and that it can contribute to confusion, especially when we're trying to communicate clearly across different audiences. When we italicize and capitalize cannabis, we're referring to the genus, just like canis for dogs. But in everyday conversation, lowercase, unitalicized cannabis is a colloquial category, like dog. Cannabis in this way is used to describe THC-rich types and usually excludes hemp. In some regions, though, cannabis refers to any product meant for consumption, regardless of THC level. Then there's the legal terms, hemp and marijuana based on THC content. Hemp is for industrial use and has very low amounts of THC. As I said, in the US, that also includes CBD types and CBG types as well. Marijuana is typically for consumption and has higher levels of THC, generally more than 10 to 30, somewhere, more than 10%, up to 30%. But here's the problem. Cannabis, the genus, includes both hemp and marijuana. But in casual and everyday professional conversations, cannabis and hemp are often treated as separate things. The reality, though, is that hemp is cannabis. Not all cannabis is hemp, and that nuance is often lost. Humans categorize things to make sense of diversity. A shared language is essential for communication and clarity. Taxonomy, as we all know, is the science of naming, defining, and classifying organisms. In the case of cannabis, we have Cannabis sativa, which most of us now consider a single species that contains many different categories. Dogs are a great comparison for this. Canis familiaris is also a single species, but includes a wide variety of types. We got herding dogs, hunting dogs, toy breeds, etc. And we can group dogs by physical traits, but we can also group them by their job or their behavior. Or, you could call that their usage group. And cannabis can be categorized in the same way, not just by appearance, but by use, chemistry, or potentially effects. But this is why language matters, because how we classify things shapes how we understand and communicate about them. And I think of making choices like a dichotomous key that we would use in taxonomy to shape preferences. Over time, we learn how to talk about what we like. Take wine, for example. You start simple. Do you like beer or do you like wine? If it's wine, then you've got choices: red, white, rosé, sparkling, dessert, fortified. And let's say you pick white. Then we've got dry to sweet, we've got still or bubbly. And those choices might shift depending on the season, the setting, or even the food that we're pairing it with. We're constantly asking, do I want this or do I want that? And that's how we develop preferences, through experience and comparison. Even within one category, like Chardonnay, there's huge variation. Was it oaked or unoaked? Where were the grapes grown? What techniques did the winemaker use? And just like with wine, cannabis preferences are shaped by experience. And in the interest of time, I'm not going to walk through this whole slide, but you get the idea. Eventually, broad categories become more refined and shape personal preferences. And there are so many factors that shape the final product: where it was harvested, when it was harvested, how it was cured or stored, where it was grown, who grew it, and how they grew it. Spend 5 minutes on LinkedIn or in a cannabis forum and you'll see bold claims like, there's no such thing as indica or sativa. But here's the thing: indica and sativa, those terms are flawed and inconsistently used, but that doesn't mean they're meaningless. Tossing them out entirely erases the shared language of many people that still rely on those terms to describe their experiences. Strain names, they can be messy and unverified, but they're still how people communicate. We name what we care about, and cannabis is no exception. Marijuana, yes, that term has a politicized and racialized history in the US, but it also has deep roots in Latin American culture and even into Africa. It's complicated, and it's not just a word to erase, in my humble opinion. Hemp versus cannabis. Hemp is cannabis, but depending on whether you're speaking legally, scientifically, or casually, the definitions shift. Genetically, there is a separation between hemp and marijuana, but it's all still cannabis sativa. And it's not all about the terpenes, and it's not all the same plant either. It's one species with many distinct types, like different dog breeds, and those differences matter. So yeah, this is where we're at. Mixed messages, strong opinions, and a real need for aligned, inclusive language. And I spent a lot of time exploring this whole indica sativa thing. It gets people riled up quite a bit. The confusion partly comes from using the same words to describe two unrelated things: effects and morphology. You can't predict effects just by looking at the plant. And originally, cannabis sativa and cannabis indica were botanical terms based on morphology, how the plants looked and where they came from. But today, most consumers never see how that plant grows, what it looks like. And when they say indica or sativa, they're talking about effects, not leaf shape. And we hear things like sativas are energizing and indicas are relaxing, which is maybe a helpful shorthand, but not always accurate or true, because experiences differ among people. And the genomic data doesn't strongly support clear-cut indica or sativa clusters, but the terms persist, especially in retail and in the consumer world, because they're useful as bucket categories, a starting point for discussion. But one interesting possibility that I ponder is that these perceived differences in effects might be tied to persistent, locally adapted traits. For example, a plant from Thailand might have evolved to deal with intense UV light, or certain types of insects or pathogens, and that led to producing certain types of compounds that help the plants in their defenses in that area. Meanwhile, plants adapted to the Indian subcontinent with different conditions would face different pressures and develop different responses. And these pressures could result in small but meaningful differences in the genome, which in turn could influence chemical production. So while indica and sativa may not clearly map onto genomic clusters, there might be real biological reasons for some of these patterns that we perceive. And I want to bring to your attention an interesting comparison. Take the golden winged and the blue winged warbler. These are considered two distinct species, but genetically they're almost identical. They differ in only 6 small regions of their genome, which is just 0.3% of their total DNA. But those few differences lead to very visible changes, especially in plumage color and pattern. Scientists believe this divergence is driven by a small number of genes related to appearance. And despite such a tiny difference, these birds are treated as separate species. So it makes me wonder, could something similar be true for cannabis? Maybe small genomic differences, especially those related to chemical production, are enough to explain some of the meaningful variation we observe, even if most of the genome is the same. So while the current data doesn't strongly support a distinct indica sativa split, I have to ask, in the genome, have we looked everywhere? In the chemotype, have we really looked at all of the phytochemicals? I don't think we have, and for that reason, I'm not convinced that we can confidently say that there's no such thing as indica or sativa, at least not yet. And this one always gets people fired up. Some folks insist that you can't use strain for plants, strains are for microbes. But I'd like to say, not so fast, in tons of peer-reviewed papers, you'll see strain, cultivar, and chemovar potentially used interchangeably. And while technically strain isn't a formal taxonomic rank, that's not really necessary. The botanical application of strain is for a group with shared traits from a common ancestor, often shaped by breeding, and that absolutely applies to cannabis. Cultivar means cultivated variety, a recognized line with consistent traits that is often registered, which often is not true for cannabis. And sure, I see the argument, if it's cultivated, it's a cultivar. But strain is a word that people know. It's familiar and it works. So really, for most cannabis that isn't genetically stable or inbred or registered as a cultivar, the term strain isn't really wrong. And I've heard tons of people say that strain names don't mean anything. And actually, some of these sensationalized headlines came from my own research, but I don't agree with this. I do think that names matter because they allow us to communicate. And yeah, sometimes they seem arbitrary, but they can also be descriptive. They can tell us something about flavor, origin, or lineage, or more. Like a name like Lemon Haze gives you a sensory cue. You might expect something to be citrusy or maybe uplifting. And sure, names get mixed up, but that happens all the time, and it's not just in cannabis. Do we need to identify and verify strains being sold match their name? Yes, and I do think that we'll get there. Apples, grapes, olives, all used to have this same problem before we developed tools for genotyping. Like the name Granny Smith, for example. You know what to expect. It's green, tart, crisp, and it's an apple. But if I hand you another green apple that is similar to a Granny Smith, you might say, oh, this is a lot like a Granny Smith, even though it's not a Granny Smith. And that one sentence tells me a lot. It gives me a point of reference, and that's exactly what strain names can do. So strain names aren't always reliable, but they're how people remember what they liked and communicate what they want. Names can hint at parentage, breeder, flavor, or effect. But we need to back these names up with real data such as chemotype and genotype. Because unlike puppies, cannabis can be cloned. And that means we can aim for consistency. But even if we have messy polyhybrids, kind of like a litter of mutts, we can still give them names. So why shouldn't we name our strains that result from phenohunts? And these naming problems aren't unique to cannabis. There was a molecular biology class, I don't remember where, that did a DNA barcoding project. The professor sent the students out to collect fish samples from the sushi restaurants and grocery stores. The students brought back 9 samples for testing, and it turned out that 7 of those 9 samples were mislabeled. And studies show widespread mislabeling in this industry. 20% of seafood samples across 55 countries were mislabeled, and 44 fish products in Canada were incorrectly labeled. Why? Well, most people never see a whole fish. They just see the fillet. So you can really slap any kind of name on it and people wouldn't know. And it's kind of the same with cannabis. We see buds in jars or in packets. We don't see the plant. And clones can get mislabeled. Names can get passed along wrong, sometimes by accident, sometimes on purpose. And right now, we don't have a consistent and accessible genotyping or verifiable identification method for cannabis. So here's a grocery display where the fruit was labeled as curved yellow fruit or long yellow thingies. but they already have a name. They're bananas. But for some reason, in these two places, they came up with something descriptive to label them instead. Renaming and mislabeling happens in many industries. Here's another grocery store. This is not a kiwi. This is not a watermelon. And that's definitely not milk. And the same goes for the container labeled cherries. Those are limes. So it's not just cannabis. We can keep the names. It'll all work out. We can also categorize cannabis by genotype based on its chemical profile or aromatic traits. This study looks specifically at the exotic aroma spectrum, and the researchers were able to identify a wide range of aroma descriptors. And you can maybe see some of these along the bottom, things like sweet fruit, banana, into chocolate, floral, lemon, blah, blah, blah. The spectrum goes from sweet descriptors to prototypical aromas and then to savory aromas. And they came up with the molecules that lend to these aromas, and they're not just terpenes. So this graphic shows 5 siblings, all from the same seed lot, but with a wide range of aromas, from sweet, which is a sweet phenotype, which is up here, and then going to the most savory phenotype down here. And what's surprising was the differences aren't due to terpenes. These heat maps are Pearson's correlations. The dark colors indicate similarity, while the light colors indicate differentiation. On the left, you see the terpene profiles, and they're all very similar. But on the right, you see the heat map that shows major variation in the aroma. SB1 and SB40 here are very similar, and SB1 and SB39 are not. And along the bottom, you see the terpene profiles of all 5, which are fairly consistent across all samples. So what's behind these differences if it's not terpenes? The researchers point to flavorants as the drivers of aroma, not terpenes. So you see, it's not all about the terpenes. Competitions like the Emerald Cup use flavor and aroma to categorize entries. For example, this is the Jackson Hayes class, which is mostly considered sativa leaning, using that word sativa. Each category has tasting notes like fruity, pine sol, and haze, in this case, the reported effects, and a list of common cultivars in that class. And accompanying this are terpene profiles. But if you look closely, these terpene the terpene data doesn't always align. These 2 samples, one has a lot of myrcene, the other one does not. So even though they're categorized in the same way, the chemotypes don't fully match. Next, there's the OG and gas class, generally considered hybrids. The aroma descriptors include gas, fuel, sweet citrus, and pepper. The reported effects range from uplifting and stimulating to analgesic and relaxing, which honestly are a bit contradictory. Again, we see discrepancies in the terpene profiles of these samples, suggesting that terpenes alone might not fully define these classes. And then there's the exotics class, which is made up of rare or unusual terpene combinations. And there were more classes. These are just the 3 I chose. In this case, the terpene profiles are even more variable, and some don't match up at all. And this category is built around novelty, not consistency. So these categories are helpful and they reflect real consumer experience, but they don't always align neatly with the terpene data. That tells us something important. Effects, aroma, and chemistry are each part of the story, but none of them can fully define cannabis categories. Style or flavor-based groupings are based on how people describe cannabis, how it smells, tastes, and feels, but those descriptions don't always match up with the major terpenes identified in lab results. So yes, flavor categories and chemical clusters are useful tools, but they're incomplete when used in isolation. And here's an example from my own research. On the top here, I'll just use the Durban poison example. These are all genetically identical, verified. They came from different cultivars or cultivators, different dispensaries, and presumably different environments. And as you can see, there's substantial variation in the terpene profiles, despite being the same genetically. And this is this similar variation in the terpenes from samples sharing a genotype in OG Kush, Mob Boss, and Blue Dream. They're showing a similar pattern. And this tells us that environment has a major impact on the final phenotype, on what the consumer actually ends up purchasing. So even with a consistent genetic identity, what shows up at the counter can vary significantly depending on how and where it was grown and treated post-harvest. And so here's how I see the current state of cannabis communication. We've got 3 groups, and they're all speaking different languages. Scientists use technical terms like cannabis sativa, italicized, of course, genotype, chemotype, type 1 through 5, broadleaf and narrowleaf drug types. Industry talks about cannabis, lowercase, unitalicized, THC percentages, major terpenes. And then consumers they care about indica, sativa, strain names, and effects, and that's their language. So each group is categorizing cannabis in their own way. And where those categories collide is a dumpster fire. But categorization is human. It's how we make sense of the world. The problem isn't that we're classifying cannabis, it's that we're doing it without alignment. So no single system is capturing the full complexity. And that's why we need a hybrid model, one that balances scientific accuracy with real world usability. And then we can all move forward together. I kind of feel like this is where we're at right now, a simplistic view of cannabis that started with indica and sativa. Then we moved on to THC and CBD driving the car. And now we've switched gears to terpenes. So it's progress, but I think it's still oversimplified because two plants might look identical and yet be experienced completely differently. And there's still much we don't understand. And those of us in genetics should be digging deeper. Morphology isn't really telling us much anymore, especially with all of the hybridization going on. But people are still reporting meaningful differences in effects. And I think there's something there, maybe residual traits from those local adaptations. We've moved beyond just THC and terpenes, and now aroma science is pointing to things like thiols and sulfur volatiles that might be doing more than we realized. It's exciting. Let's go after the hard stuff, such as subtle genomic differences, overlooked compounds like the Tropicana sulfurs, and others that are just being discovered. So just to wrap this up, even if we can't agree on this, or if we can't agree on this, who can? And here's where I land on my soapbox. Terms like indica, sativa, and strain names, they're not scientific, but they're how people talk about cannabis. So let's meet them where they are. Science hasn't settled it either. We've got genetics, we've got chemotypes and effects, but we're still figuring it all out. So how can we as scientists tell people how they should talk about cannabis when we don't even have the full picture yet? If we want to build trust, we need to align science, industry, and culture and stop dismissing legacy language. Instead, let's use data and transparency to figure out where this language came from, how it's being used, and how we can build a system that's accurate, trustworthy, and usable, one that people can actually navigate. And let's build that together. And if you want to dive deeper into a lot of the topics I touched on today, Daniela Vergara, who's talking later today, and I contributed to a chapter in the new book, The Cannabis Genome, a new era in genetics, genomics, and breeding. And the introduction of the book concludes by saying, this book is a call to action for researchers, policymakers, and the general public to view cannabis through a new lens, one that it appreciates its potential to improve lives and drive scientific progress. Let's move forward with an open mind and a commitment to uncovering the truths that lie within this extraordinary plant.